Hey guys, welcome to Psychology Spirituality. Today I'm speaking with Shadab, who studies neurocognitive behavioral science. Maybe I'm getting that right. Maybe I can just let you. I can just let her do the introduction.
Hi everyone, my name is Shadab. I'm a third-year graduate student at University of Houston, and my plan is to complete my PhD in Behavioral Neuroscience at the Department of Psychology.
Okay, so maybe you can tell us a bit about what behavioral neuroscience is.
Sure.
It's a big term, I don't know.
Yeah. So neuroscience is basically studying of brain. Behavioral neuroscience is studying of how individuals' brain affecting individuals' behavior. Basically, it's a relationship between the brain, behavior, genetics, and environment.
Okay, very interesting. So I know you guys work at a lab here. Maybe you can talk a bit about what your lab specializes in.
Sure. So my lab is interested on how substance use disorder influence brain and behavior. Basically is interested in developing and testing of novel pharmacotherapies for substance use disorder. And one potential target that we're looking at is gut microbiome, which is often referred to as a second brain.
So we wanted to do something a little different for this interview. Usually we just have a back and forth, but Shadab has a presentation that she's presented before. So we wanted to actually show you guys the slides to make it a bit more visually engaging for you. So yeah, we can get right into that. Yeah.
Sure. Target, as I talked about, gut microbiome, which has been a great topic of interest recently in the neuroscience. I will talk about my specific research interest in the gut microbiome and our plan to investigate how methamphetamine use impacts the gut microbiome. I would like to start with defining some terminology. Microbes defined as tiny living microorganisms such as bacterium, fungi, viruses. Microbiome refers to the full components of microbes, their genes and genome in a particular environment. And microbiota refers to the microorganisms that live in an established environment, such as gut. The dendritic nervous system, the part of the nervous system that is tasked with controlling the gastrointestinal system, is often referred to as our body's second brain. This network of neurons in the gut is as plentiful and complex as the network of neurons in our spinal cord, and also it has to listen to trillions of microbes residing in the gut. Our gut microbiota contains hundreds of trillions of microorganisms, including at least 1,000 different species of known bacteria with more than 3 million genes, 150 times more than human genes. There are 10 times more bacterial cells than the number of human cells. However, this is debatable. A recent study has shown that the ratio of bacterial cells to human cells may be actually 1-to-1 ratio. Therefore, understanding this hidden half of ourself, our microbiome, is essential for understanding of related diseases and physiological states. We already know that our gut microbiota offers many benefits to its host through harvesting energy, protecting against pathogen, regulating host immunity, and many other things that there is a potential for these mechanisms to be disrupted as a result of altered microbiota composition, which called dysbiosis, which may be due to illness, diet, antibiotic treatments, and drugs. We often talk about gut feeling when we meet somebody for the first time. We're told to trust our gut instinct with making a difficult decision, or it is gut check time when we're faced with the situation that tests our nerve and determination. So the idea of connection between the gut and brain function is surprisingly old and is not just metaphorical. Our brain and gut are connected by an extensive network of neurons and a highway of chemicals and hormones that constantly provide feedback about how hungry we are, how stressed we are, or if we ingested a disease-causing microbes. Therefore, this bidirectional signaling between the enteric system and central nervous system is called the brain-gut axis. It's becoming increasingly recognized that psychiatric and neurological illnesses are often comorbid with GI pathology, including Parkinson's disease, Alzheimer's disease, autism, stroke, anxiety, depression, chronic pain, and drug addiction. For example, 70% of autistic patients experience gastrointestinal tract-related symptoms, such as diarrhea, abdominal pain, constipation, and others. Moreover, autistic children have a significantly higher content of specific bacteria in their gut compared to the normal children, which a study in mice show if we place that specific bacteria, inside the normal mice, it will develop autistic behavior. Several experimental approaches have been used to study the modulatory effect of gut microbiota and gut-brain interaction, such as fecal transplanting of the fecal bacteria from a healthy individual into a recipient, or using germ-free animals which have no microorganism living in or on them. Germ-free animal studies are used to evaluate the impact of microbiota as a whole upon stress and cognition and central nervous system development. Some other studies have used specific pathogen-free animals that are free of a specific list of pathogens by routine testing. And there are studies that have used wide spectrum of antibiotics that affect the microbiota composition, or using probiotics. There are living microorganisms which, when administered at adequate amounts, could promote intestinal health and improve behaviors associated with stress-related conditions. Let's talk about gut microbiota and behavior. For the last 10 to 15 years, there has been increasingly scientific interest in the gut-brain axis, in particular how gut bacteria may influence the brain and behavior. In fact, there are several recent rodent studies that have identified the importance of gut bacteria in blood-brain barrier permeability, hippocampus neurogenesis, and microglial function. In addition, several studies have shown that the changes in gut microbiota could produce anxiety and depressive-like behaviors, as well as affect performance on memory tasks. Our lab is interested in the potential influence of gut bacteria on depressive and anxiety-like behaviors, given that depression and anxiety are some of the symptoms a chronic methamphetamine users could experience during withdrawal. Let's talk about gut microbiota and substance use disorders. Substance use disorders is a recurrent use of alcohol or drugs that cause clinically and functionally significant impairments. It is a chronic relapsing disease characterized by compulsive alcohol or drug seeking and use, and accompanied by functional and molecular changes in the brain. These substance use disorders could be from alcohol, opioids, tobacco, psychostimulants such as cocaine and methamphetamine, and others. And there are no approved pharmacological therapy for psychostimulant use disorder. For example, studies in rats and humans have found alcohol caused bacteria overgrowth and dysbiosis, and have shown chronic alcohol consumption induced long-lasting dysbiosis, triggering intestinal inflammation in humans. So for my research specifically, I'm interested to know, does chronic methamphetamine administration significantly changes the gut microbiota in rats? And if it does, how does methamphetamine withdrawal impact behavior and gut microbiome in rats? Met is a psychostimulant drug and a Schedule II stimulant, which means it has a high potential for abuse. According to the United Nations Office on Drugs and Crime, methamphetamine is the second mostly widely abused drug. So we're hoping to be one of the pioneers in understanding of methamphetamine use disorder and microbiome connection.
Thank you to Shadab for sharing her presentation with us, and if people are interested in learning more, I have some links that people can click on in the description below. We will see you all in the next episode. Thank you.